So while the speaker is getting ready, I'm just giving an overview and overall of the second session part. This goes in the morning. We hope to talk about standards and how data exchange were, the formats, how to capture metadata, and try to get your feedback. How is the best way to do? Really, this feedback part and telling us how to better do these things is the key that we are after. So these workshops really is too short to be able to really train something on hands-on and be able to use it, but it's sort of each one, each group, each work is a plenty there and plenty of people that we really didn't have time to explain. But we're trying to really point, see these are the places that to work on. So I guess in the second part, we see how this links into journals, how this, and then more on data repositories. So there will be a session on Metabolite and a session on the NIH Workbench. So it gives you a bird's eye view and overview. So I try to now pass it on to Susanna, if you please. Start, there will be 3 Nature Scientific Data and Giga Science journals, so please.
Can you hear me now? Yes. Brilliant. OK, I'm going to give a quick introduction of scientific data, which is a new open access data publication by Nature Publishing Group, and also give you an example of our first Metabolomics data set being published in this platform, because we only launched a couple of weeks ago. So I work at the University of Oxford, and Philippe Roccasera, who presented before, it's my colleague, so I'll be also working on the ISA project. But I'm also a data consultant for Nature Publishing Group, so my talk is going to be on the scientific data. Right, so this is just an introduction slide. This is some very nice infographic that I took from Digital Science, which is a company part of Macmillan, so the sister company of Nature Publishing Group. And this is quite nice because it simply shows the problem that I guess we are fully aware of, the fact that there is a lot of data. Data is growing. Significant investment is made in producing this data, yet a lot of the data get lost, or it's not accessible, or it's very hard to be reused, or actually, it's very difficult to verify if the data is correct, and so on and so forth. And as you know, many data funding agency, journals are developing data sharing policy, data preservation policy. So everybody understands and recognizes the need for preserving all this data, which has been actually funded and produced. Of course, journals are part of this ecosystem of stakeholders that realize that they need to look after the information. And of course, journals have been publishing the narrative, our discovery, the hypothesis in our discovery. But now, in a very data-centric era, they are also changing. The way they think about publishing information is changing. So of course, I mean, this is a screenshot of the paper from 2001 when the human genome was first released, and it was like a big, long, folded extension to the paper. But nowadays, I mean, this is just a couple of years ago when the ENCODE project was published, we talk about more digital information. So it's not just about publishing the narrative, but it's complementing the narrative with all the digital, all the data, and everything else that supported the conclusion and the hypothesis. Okay, so of course, publishers, as I said, are thinking that there must be new ways of storing the data or helping research to store the data, preserve the data, share the information, and make it reusable. Now, in Nature Publishing Group, there is a lot of activity which are going on in terms of how can they better deal with data. For example, even the source information behind the figure are being made available, so the number are made available. There is the concept of extended data. There is a concept of data citation. They're thinking about how the code, the algorithm can be also reviewed, because of course these are the things that produce the results. And they put out at the end of last year, I believe, a reproducibility checklist. So really try to totally understand what are the elements that need to be reported when publishing an article where we're talking about the discovery. And they also launched a linked data publication platform to disseminate information which is part of the article. So it's a lot of activity, but the activity I will focus on, it's an experimental platform, which is scientific data. So Nature Publishing Group has been working for a couple of years on this idea of how to become a player in terms of data preservation. And that's actually somehow the output of it, scientific data. So first of all, I just want to say that we have an internal curator and we have a growing curation team which will help in managing the information. So it's slightly different than the typical journal that deals just with the narrative. We also have an advisory panel on top of the editorial board panel that also includes funders, librarian, curator, not just a researcher, because it is not just about reviewing the science, but it's also reviewing the data, the code underlying, and so on and so forth, which I hopefully explain as I go along. And this project is supported by Dryad, by Figshare, BioSharing, which is an activity I run at Oxford with several communities, and also by Yasato. Okay, so I'm going to briefly introduce you what scientific data and the concept of a data descriptor, because it's slightly different than a traditional article. I'm going to explain you how the data descriptor is composed in a narrative and a structured component, which is searchable. And I'm going to give just one example, which is the Metabolomics example we have, because it's relevant to this community. But because the platform is live, you can browse and see the other content that we have. So as I said, we launched at the end of March, so we are very young. The idea of scientific data is to provide credit for sharing the data, so giving credit to researcher, technologists, curator, data scientists to share the data and make it usable, so very rich description of the data. It's focused on reuse, and I'll explain that because that's very important when it comes to reviewing the article. It's about data being peer-reviewed, as I said, but also curated in-house, and I'll also explain you how. But we also promote community repositories. So this is not a database. We work with databases, and I'll explain you how we do. And it's an open access platform. We we focus on life science, environmental, biomedical science, but we're not limited to it. So we'll expand in other areas that MPG covers. So this is just a screenshot of one of the data descriptors, but really I'm using it, and actually this is from Japanese authors. I'm only going to really use it to describe that there are two components, of the data descriptor, which is the main content and article type. So we don't have review, we don't have commentary, we only have data descriptor. And occasionally we have invited an editorial to describe a certain data descriptor or certain topic. So the data descriptor contains a narrative component, which is the typical PDF, the HTML, the readable part, but then also contain the experimental metadata, which is the structured component, which is curated and annotated with ontology as relevant. And that's the part which is in ISA format. Okay, so dig into the narrative components. These are actually the section that the narrative components contain. But the reason for showing the slide is to show that in reality the core, the emphasis, it's on the reuse of the data, therefore on the technical validation, the record, how the data can be used, and what's the method to produce the data. And this is traditionally the section for which you have very limited space in a traditional article. So the focus, as I said, it's on the method, on the technical analysis, and the quality of the measurement. And a data descriptor does not contain hypotheses. And I'll explain you how this complement a traditional article. In the narrative components, we also link to the data file, which, as I said, are in specific community repository. So there is the narrative part points to specific data files and link to these data files in appropriate repository. The structure component of the data descriptor, it's in ISA format. So you heard already from Philippe what ISA format is. So I'm not going to go into any detail. So the ISA format is it's the structured representation of the data descriptor. We'll have information about who has done it, what type of experiment it is, who has funded it, how many samples, what the characteristic of this sample, what kind of technology you use to report which type of measurement, where the data file are, and so on and so forth, pointing to the data file, to the analysis method, to any script or any code that has been used into the analysis phase or in the data production phase. So the ISA file, as you heard already, can be customized depending on the sample you have, on the technology you have, and certain ontology can be used to tag certain terms to describe the experiment, and the convenience of using ontologies because it will facilitate the data query, data comparison across study. What we have, it's an internal curation team which actually assists the author to create the structure. So we know that's not a trivial thing to do. And there was a comment before, it's very complicated to create any structure for a researcher because you are not used to. You are used to create an experiment, but you don't know how to actually represent information. That's fair enough. So that's why we have an in-house curator which will guide you via a set of templates, via an online tool, to create and tease out the information that we need to represent in a structured manner to make this experiment machine readable and later queryable too. What we have also done with scientific data, and Philippe helped in this work, that we created a specific specification of the ISA format for scientific data. Because, as I say, it's customizable, and you can add also specific fields for later drive the query, we really spelled out all the fields that we want when people submit to scientific data. And we created this data with this specification document. Now, this document, it's not for researcher, but it's for service provider, tool developer, anybody who wants to automatize the export of this ISA tab format for submission to scientific data. That's what that's for. Really? Oh gosh, I'm really, really late then. Okay, so we need to make the data discoverable. The idea of scientific data, it's at the center of, it's between the papers and the databases. So that's why the database we're here will contain the data files that we point to. The scientific data will contain the ISA tab file, and the journal will contain all the narrative, the hypothesis, and the discovery. So we help author to search for the right databases where to store the data. And the point, obviously you work in Metabolomics, so you know which databases are for you. But because we cater for a lot of data producers, we actually have to make a list of these databases and what they are and which one we endorse. And for this reason, actually, we're using BioShare to catalog the different databases. And Metabolite, it's one of the databases we recognize. And there are several other, over 50 of those databases. Now, the question is, how do you actually relate a traditional article with a data descriptor? The traditional article, which is here, it's about the scientific hypothesis, the analysis, and the conclusion. The data descriptor, it's about the method and the technical analysis supporting the quality of the measurement. This means you could even submit a data descriptor before you develop and you submit a traditional article. And we have an MPG pre-publication policy for this. You can submit a data descriptor at the same time of your article or after. And we have cases of all the three in our actual content, where you can add further information for it. Okay, so are we going to peer review the data descriptor? I'm going to go really fast. So although this is very detailed, this is from the website. And all I'm going to say is that the evaluation of data descriptor is not done on the perceived impact or the novelty of the finding, because this is not about discovery. It's about data reuse. Can this data, can I reuse data? Is that useful for somebody else? So there are very specific criteria which we divide in three, and we guide and help the reviewer to understand these criteria and make the reviewer in this context. Okay, so the current content. The current content, it's really varied. We have from neuroscience to epidemiology, functional genomics, and of course we have our first metabolomics submission. Our data sets are in different repositories, so you can see data set linked from different repositories, and those number of repositories will grow as we have more submission. We even have code deposited in Figshare in GitHub. We have little data, smallish data, depends. We have individual data set, but we also have compendium of data sets. You don't have to submit necessary individual data set, and we have several citizen science projects. Okay, this is actually the example. I only have final slide after that. So this is the example of the paper from Mark Vyant for Ralph, which is in the audience, and colleagues. So this is our first metabolomics data submission. And actually, you can see that the data file was along with, which actually represent all the experimental steps, all the processing steps here. It's deposited in Metabolite. So you can access the PDF, you can read the description of the experiment, and then you will have the link in Metabolites. Now, this is a curious example because both scientific data and metabolites use the same format, the isotype, but the one in scientific data is richer. Sorry, the one in metabolites is richer because it's very metabolomic specific. We'll have way more information than the one we store in scientific data. And my final slide, it's if you are interested in submitting or collaborating, and if you run a data repository which you think scientific data should recognize, please do have a look at the criteria, or how we recognize repository, or come and talk to me. If you're interested in submitting and you are a tool developer, a vendor, or a database system you would like to support in this specific configuration for helping your researcher to submit to scientific data, there is the isotype specification, and again, I can help you with questions, and so can Philippe. If you want to submit data descriptor, you can even also ask us, because it's a quite novel concept that some people don't necessarily understand. People have said description of databases. That's not what we get, but we could get compendium and aggregated data. So if you are in doubt, do contact us, and we actually can tell you and can guide you in the submission process. Thank you.
I guess we go to the next speaker for interest of time, and then after two of them, they can have a question if there is.
So we're going to tag team on this, and I'm going to cover data, and Rob will cover more workflows and methods. And Susanna's done a great job kind of setting the scene for data publication, so I'll try not to repeat too much of that. How does this work? Is this? So yeah, ultimately, I'm a big fan of this quote that articles are merely advertising of scholarship, and the stuff that people really need is the data and the computational methods. But until now, there haven't really been, but all of the focus has been on this advertising. There's been a lack of credit for anything other than that in traditional publishing models, and it's kind of held things back in a way. And this is a big deal, really. Like topical being here in Japan now, this big acid stem cell scandal continuing to continuing to cause problems, and a lot of this is because of this the incentive structure, the fact that protocols were not released until 3 or 4 months after the original paper. There's been a lack of supporting data, code, and things like that, and it's just the incentive structure ultimately can result in these kinds of things, lack of replication studies, no incentives for replication studies, and things like that. So it is really important. We can really see this now. It's very topical, and it's unfair to just focus on this one scandal. It's everywhere in the world. There's this increasing number of retractions. In the last decade, it's gone up 15 times, and there's a great calculation from Bjorn Brems that at the current rate of growth, by 2045, there will be more papers retracted than published. So we need to. This is important. We need to address this. So Susanna, has done a good job showing that there is, you can now incentivize data release, and we'd like to use similar things for software, peer review, all of these kinds of things. Give credit to these other really important things we're using this data site infrastructure. And so this is what we've been doing at GigaScience. It's a project hosted at BGI Hong Kong. So we're based at the BGI, working with the open access publisher Biomed Central, and so we have not just publishing platform, but we got a data publishing platform, and also analyses and other kind of infrastructure that we can use, being based at BGI. So on top of regular papers, here's a nice one that Razar is on. So one thing we're doing slightly different from the scientific data approach is we actually have a data publication platform as well. So there are, on top of all of these other databases and repositories, we can host. We have the ability to host things that don't necessarily have their own repository. We've got this infrastructure at BGI that can host these very large data sets and give them DOIs. So this is GigaDB. We've got about 120 data sets right now, and it's very focused on big data. So we have a Spera download that's 10 to 100 times faster than FTP. We've got this very multi-omics focus. We have a lot of genomics data sets, but we've got proteomics, lots of imaging data, and we've been working on metabolomics data recently in the hackathon that people were talking about previously. We've also been working, with ISA, and our curator can help with deposition with, if there are, repositories already out there. If you want to know more, we have this paper in database recently. So this is in data publishing, this is our kind of focus here, but we've been trying to take things beyond that. Ultimately, these are the kind of people that that can benefit through this data publication. These are rice breeders, and last month, timed for International World Hunger Day, we published 13.4 terabytes of rice genome data. This quadrupled the amount of data in the public's in the public domain to basically ultimately aid rights breeders. This is all Gates Foundation funded, and they've built a Galaxy server and trying to build cloud resources. So to really kind of democratize this kind of research that you don't need big computational power. So then the next part I will hand over to Rob, who can tell you about some of the other tools we've been building.
Thanks, Scott. Yeah, right. So I'm just going to talk about two of the new tools that we've been developing to help share and publicize, help you to share your data and your pipelines. So moving on, I just want to bring back to this slide. So we have our GigaScience is our open access journal. We have GigaDB that Scott talked about, which is our data publishing platform, and I'm going to mention how we are now moving on to publishing images and allowing more easy access to images via that. And then we have GigaGalaxy, which is our Galaxy platform through which we publish tools, software, pipelines. So moving on to GigaGalaxy first. As I say, this is where we will take your tools, your software that you make, your pipelines that you've built up over time, and we will publish them and give them a DOI that then makes them citable, shareable, reusable, hopefully giving you some impact reward from them, but also just helping the community to build up more robust pipelines. We have a live server here where you can go and play with the tools that we have. I'll just go on next, yep. This is, for anyone that doesn't know what Galaxy is, it's an open source platform. It has tens of thousands of users registered on the Galaxy project's public server, but it has about 55 public servers active at this point in time. There are over 1,000 papers citing Galaxy. So it really is being taken up by many, many people. It's designed for biology, for mixing biological data analysis tools, but it is being picked up by other fields as well. I'll just move on. This is what Galaxy looks like when you have it in operation. It's as simple as could be. On the left-hand side, you get a list of tools. So these would be tools that you've put into your own Galaxy server. There are a host of tools, thousands of tools that you can download from other people's servers and just populate your own tool shed with them. When a user clicks on a tool, they are presented with all the parameters they could possibly fill out, and when they press play on that or execute, they build up a history on the right-hand side, which is their pipeline. And then that can be saved, shared, downloaded, passed around, etc. So it's very, very, very intuitive interface, and very simple. Here's a relevant example that we're just in the process of finalizing and publishing. This is the Viant Group in Birmingham has been developing their Metabolomics Galaxy. And this situation should be quite familiar to a lot of you. So we have a legacy system that is all developed in one programming environment, in this case MATLAB. And that was quite a good system. It goes all the way through to some statistics there, the whole 9 yards. But then you have different spectrometer methods that you want to bring in, so you start using a different package. But then that doesn't do everything you want, so you merge it into some of the existing tools that you have. And then you get a PhD student or something in who develops a lovely package in another programming environment. And so you end up with Python or MATLAB, lots of different toolboxes and quite a complex workflow. And many people in your group may never have used Python, or maybe they've never used R, or maybe they've never used any of them. They're not necessarily programmers. Well, the thing about Galaxy, for example, and other tools like this, it makes it very simple for the user. So you take your original tools, you create a very simple Python script and an XML descriptor form. You feed this into Galaxy itself, which is a Python program itself. And the user sits there on the desktop and interacts with that very simple web form that I showed you. Knock-on effects are, you've got more secure central store of data. It's much easier to upgrade and update your workflow without having to worry about the Wild West, people not upgrading, working on old versions, etc. So you get much more control if that's what you're interested in. But in general, it's about taking all these different modules and just putting them together in a very simple, easy-to-use format. Oh, ignore that. And this is what you get at the end. So this, Galaxy is very good for visualization. This is an example of one workflow being nicely visualized. And you can click, in the web format, you can click on any of these tools that you've used in your flow. And on the right-hand side, you're presented again with your parameters that you actually used. You can save this, you can pass it around, other people can upload it into Galaxy, see exactly what you've done. They can change parameters, and they can press play again. It will run through, etc. So it's very, very reproducible. And so with the Birmingham pipeline is one of the first raw format all the way through to PCA and everything, pipelines that we're aware of in this form. So yeah. So as I say, the Birmingham pipeline is just being published with us. We have another example of this reproducibility is the SOAP de Novo 2, which is a genomics tool that we already have on our server. And if you have the pipeline, which is available, and you have the paper, which is also available, open source paper, then you can go onto our server and you can repeat the pipeline. You can get a result out, and I don't know if you can see that, but this result matches what's in the paper. So it's just trying to make published work more reproducible. That's quite a good example. So moving on to another tool that we've been developing, or we're currently developing, is image data. So we have this GigaDB, this database, where we will host your data sets, and we'll give them DOIs as well, so that they can be cited on their own and shared, and hopefully you get more impact. But what we've started getting is, someone will say they have, they've done a genomic analysis, but they've also, in order to get the taxonomy right, the taxonomic description, they've started doing micro-CT. and building 3D models and ultra-high resolution taxonomic descriptors of the organism that they used. And we wanted to be able to give full access to that. We didn't want to just host the images. Some people might not have the appropriate DICOM interpreters or whatever to interact with image formats. So we looked for a solution, and that is Omero, produced by the OHM consortium, led by Jason Svedlow in Dundee. Omero is another web server package which we're integrating with our website, and it has this wonderful bioformats part of it, which means it basically allows you to interact with about 95% of all known scientific image formats, and they're constantly updating it, and so it's really flexible. And what it allows you to do is you can not only view the images from your slices or whatever, but you can do various measurements, filters, etc. And so it's quite an interactive way of viewing these data formats without you having to download anything yourself. And we're building this so that you would be reading an article, and you'll go from the article to the data, you'll follow the data to the images. And so it should make the reading of papers give you much more access to the full depth. What we're hoping is that we'll be giving you access to all the image data, not just the cherry-picked best examples, which is very common. And at the end of the day, you'll be able to download and reprocess this data if that's what you wish to do. So again, we're aiming for reproducibility. Our version is in production at the moment. If you wanted to see a really good example of Omero in action, JCB, the Journal of Cell Biology, has their site up and running. And so this is an example of them using it to show lots of the content from high content screening, sorry. And yeah, they just get to show you everything. You don't have to look at everything, but you can get down to everything if you wish. Yep. With the images particularly, but I guess it applies to everything we're talking about, is that the alternative to this kind of openness and interactivity is the look but don't touch approach, which has these very famous retractions associated with it. So in summary, we're going to say reproducibility is very important, and currently it's not very common. This has been mentioned by various people in various of the workshops today. There are many tools appearing. We're trying to develop some, but there are plenty of others. Many tools appearing for publishing your data, your images, your tools, your workflows, and for sharing those. The incentive is data publishing gives you more propagations, but it contributes to more publications as well. You get more citations and more impact. And then because this is a workshop, we want to invite sort of discussion with the audience. And so our question is, are you convinced, or do you think that it's too much work for no reward? Are you worried, like, what are the barriers to submitting your tools and your pipelines to somewhere like us, or just putting them up on any of the Galaxy, for example, Galaxy toolsheds that are available? Is it that you're worried that your code standards aren't quite high enough, you've not commented them well enough? Are you worried that, shy about the data being maybe picked over and someone commenting on it? But anyway, so yeah, that's our sort of, are you up for it kind of thing. And yeah, so help us make this happen. Give us your data, your papers, your images, your pipelines. You can publish any of these as notes with us. All our publication costs, we're an open access journal, are currently covered by BGI until 2015, so it's a free open access journal at the moment. So please get in contact with us if you want. And that's just the people that we work with, BGI, Gene Bank, China University, Hong Kong, etc., etc. But thanks.
I guess we have a very little time, really, for one question. If anyone has from any speakers so far about nature scientific data, or Scott or Ron about GigaScience and GigaDB, please. They're happy with everyone. Okay. Tim.
This is a question for Susanna, probably more, but perhaps you guys might have a view on it as well. But I noticed, I noticed that you mentioned that scientific data will publish computational data sets as well as experimental. And I'm just wondering, you know, what do you mean by that? So perhaps you could elaborate how, you know, do you publish simulated data or what kind of computational data?
Thank you. People initially we were focusing on wet labs, you know, experiments. Okay, you have sample, you do something, you produce it. But maybe you actually are reanalyzing the data. Yes, you can develop a model, you can do some new novel algorithms. So we are approaching that. We are accepting that type of data description too. That's what we meant.
Question towards the GigaScience journal. So what happens if in my workflow between step 5 and 7, step number 6 has some human interventions? So what I've done for one of my studies is that I have a workflow script actually in R, and then the intermediate manual intervention was to manually annotate some metabolite identities. And then I kind of merged that at a position where it should be in the overall result table, but that is kind of fragile. So how comes human expertise into the workflow?
Yeah, I mean, certainly there's a lot of human intervention still in most workflows that I've ever come across. Normally at a key stage like metabolite ID or deciding on whether your confidence interval should be that one or that one. And it would be nice if these things could be automated in the end. In terms of that, what I've seen people doing is that they break the workflow in two. You can, from Galaxy, you can download as an XML, or there are ways to visualize if it's about analyzing, say it's analyzing a scatter plot and working out if you want to remove an outlier or something, whatever. Then you can do visualization in Galaxy. You can have the results printed on screen. You can choose to manually intervene. At the end of the day, yeah, it's totally possible. You would have two parts to your pipeline, and that can be shared. It's equally as useful. Yeah, no problem.
So I love Galaxy, but my question is sometimes getting all of the parts, not within Galaxy, but just all of the different parts involved in a workflow to talk to each other is difficult. Is there any discussion about just putting entire virtual machines with all of the data onto these places?
Yes. So the virtual machine thing is very useful because environments, having your pipeline run in the environment, sometimes the environment settings are as tricky to set up as anything else. So it's very useful to have a virtual machine. And so we have...
We're reviewing one at the moment, but it's kind of watch this space.
So we will be hosting the entire virtual machine, assuming it passes the review, etc., on GigaDB, our repository, along with the code separately, along with the comments and whatever else, the full 9 yards. So yeah, that's a really helpful thing to have. There is some people don't like the notion of just relying on virtual machines because it's a black box system, but I don't really agree with that myself. But yeah.
I mean, sorry, we have to move because of shortage of time. But everyone will be around, and we can, hopefully, if there's any time left at the end, we can actually extend to more discussion. But the nice theme here is that everyone really emphasizing, and this was by coincidence, that a lot of it is on data reproducibility. Just a brief overview that sort of history of EBI, which is a lot of data repository and resources, and from genomics to small molecules and metabolites. A lot of experience in data standard, a lot of experience in repository. Only these experiments, a lot of these discussions actually is being carried out in similar field and other fields. And it's just transferring that knowledge or learning experience, make it faster for metabolomics. And it is faster for metabolomics is the key message within the slide. But going back to, this is completely by coincidence that everyone had this similar emphasis on just making the data really available. And okay, mistakes happen, but it's a natural thing. And to strong statements of not making data available being malpractice, to examples of retracted data sets that you heard from Scott. But the key is really is the reproducibility. I mean, there are immediate challenges working with instruments, metabolite ID, but I guess these will pass, as I said previously. And then to be able to see whether we can generate new knowledge or ring studies. There are several ring studies, at least 2 of them, that I know making this data available after they really work is quite nice to let other people examine, or other people set up ring experiment, whether cross labs or cross samples on different layers. But also on a data layer, you've seen this slide, and if you're in the previous talk, but this is going back on now more on metabolite, capturing and moving data from more sort of lab-based, Excel-based, to more managed, and giving the advantage that Felipe explained by tagging, working, harmonized data sets, structured data. It takes time. It's not trivial. It's expensive in terms of times and resources you have put in, and really people don't want to. But it has added benefits, and these added benefits could be that in future that you can actually use this thing to more intelligent cross-queries, cross data sets, once you have large enough data sets, to be able to see what metabolites has actually changed in cross studies, or if I want to look, for example, see similar species or different samples with similar genotype on similar equipments using similar column, whether it would be 4 or 5 data sets as such, and then be able to use and generate new knowledge. Obviously, you're very, very far away. So it goes back to Rima's question, are we there yet? No, of course not. We are very, very far away from that. But we are aiming towards that. So we set the aims low. Experience from other omics and other sciences were, okay, they accepted everything. But then end up saying that, okay, we collected this data, but this data is probably not very useful or reproducible or has problems. We try to avoid as much as possible. Still will be pitfalls, but these are things that we try to aim to. This is, again, trying to make familiarity. Part of these things is collects control vocabularies. This is the term preferred, for example, to ontology. The way the description works, best thing is to give it by an example of that. This sentence is very scientifically sensible. All of us, we could read it, understand, but probably very not machine readable. So try to make that accessible and see exactly what we meant or exactly what a disease was, is the way that we try to classify. And then this information becomes more structured. So the computers can understand. Then we can use these tools once they're in place, once they're set up, to be able to look for things and look for matches. So, but it requires work. I'm not saying that it's easy. No one's saying it's easy. The expectation is to be easy, but it's not. For many years, however many years, you spend time, collect data, accumulated data, process data, handle data, submit data, and you're in the last day of publishing and you want to publish it, and somebody says that, oh, make this data available. The last thing is that, I have to now do that. How much does it take? 1 hour, 2 hours? No, unfortunately, it takes longer. So we really hope out of these talks to prepare you. Why not do it right at the beginning? Just be prepared for data submission. Why you want to leave it to be last thing? So we can actually use, there are a couple examples on lab. Unfortunately, the data is not yet public, but it becomes public soon, that they are using the same configuration right from the beginning to be able to capture data and using their own configuration file, using our configuration files, and working with ISA teams and others to creating specific need for it. You can use other tools as well. And these are, again, based on existing criteria. Again, this is another way to actually work with Metabolomics Society. Metabolomics Society being really active and promotion. There are subgroups, committees. They're trying to reignite all this work. So it is a time now to engage and get your feedbacks. Somehow make it accessible, reachable to any people that this is missing. If it was there, I would have used it. But trying to improve these things, some of them has quite a lot of criteria. Maybe it fulfills your need, maybe it doesn't. But we need essentially your feedback to see how good it is. And we have to really think about the thing that we have to provide is the job of a shoemaker, but we only can make for one size, so this size has to better fit all. These kind of analogies, whether it's correct or not, but there are so much variety in instruments and technologies and specificity. We try to be as compatible as possible, but obviously there are always going to be some odd shoe size or case that it doesn't fit, so try to tolerate us and work with us. This slide is old, it hasn't been there, but anyway, this is sort of getting into the repository side of data, and it is good that I hear a lot of people are now quite familiar with metabolites. So from the past years or two that we try to advertise and say this repository resource is available, to now more people are aware of. We've got actually a longer list of people registering, maybe 3 or 4 times more than people actually submitted. So at least people are registering. Whether they're submitting yet or not, or planning to do, is the question. So it is we're trying to engage, saying that, what has stopped you so far doing it? And we give time, discussion, hopefully another 10, 20 minutes, that what has stopped you, what will encourage you, and how we can actually use combination of, classically says, as carrot and stick to do it. But anyway, we tried to be as accommodating as possible. So we accept most formats that comes from instruments. What is a raw format that comes from instruments? Ideally, we encourage you to convert it to one of the exchange format that we talked in the morning. That will be the ideal case, because at some point we will want all of these to be exchangeable, accessible, as Stefan talked about. So, but having the raw format has its own advantage. And we not only want that, but we want to make it really reproducible. We want all the pipelines, everything we captured, all the blank samples that you think, oh, what is this important? Why do you want my blank? Because there's information there, however trivial. Or your QCs or reference compounds that use ID. There is no limitation from outside. We accept all of these things that we're asking you to provide us and make it available. So again, going back to the mantra of reproducibility. We now gradually are expanding the reference layer. So this will be on top, an addition, and linked to what's the repository, which holds the metabolomics information, but bringing existing resources tools in one place to make data, chemistry, biology, reaction pathways from chemical compounds, that metabolite or small molecules, whether it's a mass spec or NMR or anything else, gradually available, and making this accessible, as well as linking it to other resources, trying to actually cross-link data with reactions and pathways, and other information is out there. There are, I said in the morning, but I'm re-emphasis in case of people new here or watching it online or a screen somewhere, that we have all the capabilities that keep your data private for quite a long time. Usually, this will be way beyond the value and expiration date of really a study, because I don't think a study endlessly will be useful. A 10-year or 20-year study, probably no one wants to work for it anymore. But anyway, we try to work and keep it private. Within the private study, while it's there, you can still share it with your collaborators and colleagues. So these are a good incentive to work early, not just when you have to publish, but from the beginning. And there are a number of people, including Stefan, including other labs, who are actually doing it right from the beginning. There are metadata collected, but I guess the important side of it would be the protocols that are actually done, similar to your materials and method that you report. Now they are in sort of a free text format, but ideally this could be done in a way that we can actually look up and see how samples are made. I know that Metabolomics Workbench are collecting these as SOPs, and eventually between us and repository, we might be able to work in a way that collect these methods or protocols as a kind of SOP for beginners, for people who want to know how these people extracted the samples or how compared different extraction methods, maybe to the results, how did it work, and gradually collect these things to metabolites that are identified, link it to some KEBI and general information about INCHI, and any identifiable and what type of metabolites there are, to be able to look it up. Most of this job is by combination of both on the curation side and on the submitter, because we possibly cannot check everything that is correctly reported. So we try to actually work. But I guess the ideal case for us would be people who generate tools be able to do it and link their tools. So MedFrag, with Stefan's developer, MetaboQuest, or Darren Creek, we tried to work on. We still haven't given up to make his tools to work and generate metabolite IDs, and this eventually be in a nice exchangeable format that gradually we moved from our format to, for example, MZ-Tab, a simple tab-separated reportable file that not only gives a metabolite concentration or intensity or level, but actually how do you manage to do that? So knowing these things is quite a lot of work. And the platform 9 that used developed within our group for metabolomic experiments, and it does similar steps to capture steps of processing step to be able to see how reproducible they are. These are sort of snapshots of the reference compound. So I haven't updated this slide, but this number of 6,000 is now about 12,000, so it doubled the number of metabolites that is in the reference layer for each metabolite or small molecules, and link the information about species, sort of represented by like a mouse, and there's arrows for reactions, and it has NMR spectrum as a spin shape on literature search, and these are sort of linked to reactions and via another sister repository in metabolite called RIA that captures the reactions of particular metabolite. These will be linked to studies, so these metabolites that actually been identified are linked to actual metabolites, so you can actually go back and see which be able to really move cross metabolite experiment to the reference layer, as well as being able to view or download via link. Still, this is sort of work in progress very much, and we will put more time and emphasis on this thing as we get new grants and more resources and to be able to work on. This is an example of mass spec and information linked to some metadata and literature, but we hope to actually now do the next part, being able to actually provide means to interrogate data, being exchangeable format, being able to provide reviewer access to be able to see data quickly via a plugin for your browser, for example, being able to do statistical analysis. Either you want to do it right from the beginning or you want to test something, essentially creating something like a lab-based, a Google lab-based or analytic-based simple version via existing R-based or Galaxy-based, working with others to be able to make these things available from slides and people works that you already heard of that I'm not going to repeat. Have some checklists in place that actually see how good it complies with, for example, MSI, and work the main part that we sort of have funding and really looking forward to work people who actually have large lab and they want to pipeline. They already have a LIMS system or thinking of a LIMS system or adapting a LIMS system, being able to streamline these things. We are working with Biocratis, so Biocratis will try to actually work with it and be a partner and try to make their tools partly communicating, linking, so people who use that will be able to have a submission button, if you like, or submission access. We will still in discussion, so we will see how our discussion will go, but this is not limited, it's open to anyone who is interested or have a tool or application, either to promote that or use that, and be able to really link these things. This example you already seen from Mark Group, but the pipeline is really steps of different processing steps. And this is a good example that you can try and see how reproducible each step is. So it is a lot of work to do that. People usually say, okay, I give my raw and you give my Excel file that I got this concentration, but there are many steps between those two. And how you took these steps to get there, it makes quite a lot of differences. So we don't expect anyone to go through this much process of reporting and details, but it's a food for thought, really. A lot of work still required to get to that point. And in the morning, if you see the talk from Metabolomics Australia, that they're creating their own LIMS system. Sarah Barani is here somewhere, I saw. So there are actually, this is example of big, large limb system or repository resources that do pipeline for large group of people across Australia or even further outside Australia to Singapore to be able to make it easy accessible so they can have this, for example, an export function or submission function, be able to facilitate a broader barrier lower for submission. And again, I put this on another level, which is, I already showed an example in the morning, if you're there, that on repository aspect between these resources, various resources that provide metabolomics experiment, whether there are here examples of repository, metabolite, or metabolomics workbench, or GOM, and there are Netherlands Metabolomics Center, NMC, which is joining, and Mariab, which is another French data resource which has metabolomics experiments. So these are really pointers towards these resources. So the word exchange is a bit misleading because we're really not exchanging. The exchange part is really metadata. The raw data actually sits and lives in where the repository and data resources are. This is, again, a lookup, a way to see what kind of information is there on what repository. And gradually, once we can work with different data resources, repository, we bring the reference layer part of it. So not only the metabolomic experiment, but we can actually look in reference compounds and metabolites and see which new, for example, compound is acquired where, and be able to access it via some feed or announcement. So in a way, facilitate all these things via a very, very large group of people that are involved. So this is involvement of many, many, many people. It's not by all means done by us. This is part of a wider package. It's just one of the work packages by Thomas Hankemeyer Group. Michael van Wert is sort of doing the hard work for these. And this is sort of the case that I repeat from the morning, that the ideal case that in exchange format from various resources, whether it's a PSI or MSI, things jointly developed, because this format is essentially, a lot of it use the proteomics being the same instruments, and they have developed really bulk parts of these formats and libraries to be more filling the gap where metabolomics is missing, to being able to link resources and result, and be able to report and submit and get journals and others involved. And I guess these are more acknowledgements and funders, and thank you for listening. Yes, there's a quick question about metabolites before we move to Metabolomics Workbench. Scott?
I actually got a question on Twitter from Egon Willenhagen when he knew you were speaking. And he was asking, have you been talking to any journals? Is there any moves to work closely with them, potentially, start, working towards mandates?
Beginning step, one of these metabolomics journal, the official journal of Metabolomics Society, via Roy Goodacre. So we gradually, before completely announcing, we're working towards steps, and Metabolomics Society as well, as being the key driven for all these standards and these works. There are other journals, Metabolite Journal. These are all different spellings of metabolite, and Metabolite Journal is now one of the others. Is starting beginning to +1 journal is not the one that we are actually sort of talking, but it's entirely community. If you as a reviewer ask for data to be accessible from the journals that you are partly an editor or you work on, again, this has to be a community effort. I mean, it's not that we can fix all the problems, solve the problems. We're not going to solve and fix all the problems anyway, but it's just via larger community work. And GigaScience, another journal, or Giga Journal is another journal that we hope so, being on board. So question back to you. But any other people, any other journals, we are happy. There's no limitation on our side. We're just getting your time back.
Just one more comment. You said it takes a while to get your study into shape to be submitted for metabolites. So that may be true for the first time you assemble a study, but the actual steps just take for me, with some experience, half an hour. What takes a long time is actually talk to the people and ask them, is this the same aliquot from that sample? So it takes a lot of forth and back from the lab book to actually organize the data in first place to get there. On the other hand, you could say people should really be aware of where the data comes from and have that assembled in such an organization in a spreadsheet-like thing. And once we have that, usually it's that the columns are differently named and we cut and paste, forth and back, and then we have all the data. So that's what takes the least amount of work, but actually understanding the data is something. A second point I would like to make is that Philippe didn't mention in his talk that there is a web application that you can install in your own institution that will take uploads, so to speak, your own uploads of isotype studies. And that could be metabolomics, proteomics, gene expression, whatever you have. And you could use that as a poor man's in-house data management system. Because I've heard so many stories that people were actually trying to find their data after a publication where they had the GCMS data on this set of CDs and the array express, the array data was somewhere else. So if you do the actual step to do that already in-house, then the actual submission to the repositories will be much easier.
I mean, there are labs who actually gone through their learning barrier. I mean, Thomas Metz is not here, but I was really, he's submitting his 6th experiment. So they are really doing that, or Ron Weiner from Italy. They're actually 5th one. So there are, once you got past it, actually, you're absolutely right. Okay, I guess I introduce the next speaker, Padma, that will talk about the NIH initiative is another resource to make metabolomics data and resources available, and sort of an overview of the common funds and metabolomics workbench. So please, man.
Yes. I'm a program director in National Institute of Health. So we manage this Common Funds Metabolomic Consortium. And for those who don't know what NIH is and what does and how it is organized, it is United Nations Biomedical Research Agency. We fund most of the biomedical research within the country and also outside. And we are about $33 billion budget entity. And we have 27 institutes and centers, each with different mission and objectives, and mostly organized around human diseases and organ systems. And some of the like a trans NIH activities are actually coordinated by the Office of Director through common funds. I mean, this is where these common funds are coming from to support some of the activities like Metabolomic Consortium and so on. Okay. So typically, the common funds programs have to be transformative and and catalytic. That is, like they should be able to change the way we do the science, or maybe able to jumpstart some of the, like a very, fields that are in the infancy, and so that they can go forward. And also, they should be synergistic and cross-cutting, because they should be relevant to more than one NIH institute, like for example, NIH Cancer Center versus Diabetes Institute, Endocrinology Institute, or maybe like Aging Institute. So they have to be more of interest to more than one NIH institute, and also should be able to work across various biomedical fields. And they should be very unique, that such a program should not have existed before. So considering various needs of the metabolomic community within the country especially, we have come up with this program a couple of years ago. I'm sure you have been hearing about this for the past 2 years, and some of you have been really closely involved and keeping up with the progress of what we have been doing. Mainly to increase the national capacity in metabolomics and of course to train some of the new generation of scientists in metabolomics, like clinical scientists, whereas like bench scientists, and also to create some of the resources for the community at large, and of course supporting new technologies also was one of the reasons we came up with this program. And at the core we have the comprehensive metabolomic resource course, what we call them RCMRCs. They make the nucleus of the whole consortium. And other programs like technology development or training or the reference standards are very complementary to what the comprehensive course are doing, and there is a lot of synergy among these things. And to coordinate all these efforts, we have a data sharing and coordination center, also to support the international collaboration that what Reza was talking about earlier today, and also what Oliver was talking about, ring trial and so on. And if you want to learn about the whole consortium, you may want to visit the poster at Daiichi, the poster number 129, that will be tomorrow. So I'll only be talking about some of the programs that are relevant to you at least at this time, because I don't have so much time to describe everything. We have Oliver Fain at UC Davis, Mayo Dr. Sri Kumar Nair, and University of Michigan Chuck Burand, and Rick Higashi at University of Kentucky, and Research Triangle Institute Susan Sumner, and University of Florida Arthur Edison. And some of these folks are actually in the audience today, and you may have been already interacting with them. The Data Coordination Center is located at UC San Diego. Shankar Subramaniam is the PI, the investigator on this. And this is the link for the workbench, what we call it workbench. It's actually DRCC website, and each one of them has a website of its own. You can visit any one of them, or if you go to this site, you will be able to reach out to the other ones as well, and you can learn more about it. Okay, so what makes a regional comprehensive core? They are able to provide fundamental metabolomic technologies, what you are all familiar with. And so any new investigator who is not familiar with metabolomic technologies or doesn't have any enough expertise or capacity to do these things, these RCMRCs will be able to provide consultancy and also service as such. And then in addition to that, they also have some unique technologies that they specialize in. For example, Dr. Arthur Edison's group specializes in isotope ratio analysis, and then Oliver Finn, like multi-omic integration and imaging technologies and various other things. And we also support some of the technology development and early adoption. So we have, I mentioned about the initiative on technology development. So they are very well aligned with these RCMRCs. For example, Rick at the University of Kentucky, where they have both a technology development, also they have an RCMRC. So the technologies that are in the development will be able to mature into a technology that can be actually provided as a service in itself. And then we also have the training component and the collaboration and pilot and feasibility program through Outreach Core. So these are also well aligned with another program that we have for training, which is also, they provide very specialized workshops and also so like Metabolomics 101 kind of programs as well. And then collaboration. So all these services are available for free for service and also through other collaborative activities. And they also have special funds set aside for pursuing this pilot and feasibility studies so that the biomedical research community can benefit from the services that are provided here and also able to apply metabolomic technologies for this research. And the other one that of interest and maybe of interest to you as well is the reference standards. And this is actually managed as a contract mechanism, mainly to synthesize metabolic standards that especially that are not commercially available, but they are going to be useful to the community at large. And also some of the unknown metabolites that you may have some idea on what the structure is like, but currently there is no metabolic standard. So they would be able to use this particular surveys through this. And the program director I have listed here from NIH manages this program. And the RTI and SRA International have been funded, these are through contracts to make these things. So the Srinivas actually manages this. So the other one is actually data sharing coordination center that I already mentioned to you about. And this is actually a cloud-based data repository, publicly accessible. And as of today, we have more than 50 data sets. Last year, we were talking about this, that we didn't really have as much data, but we have all the infrastructure in place. But now this is the progress to date. And then also to help develop various analytical and data standards that would be useful to the community at large. And also a lot of activities that Reza was talking about in terms of data sharing and coordination is also supported through DRCC. So this is an ongoing activity and it's likely to take place, I think, over time, like as Reza was actually talking about. So currently we have a couple of things that are soliciting from the research community. One is you will be able to deposit your data if you are interested in this database. You are welcome to do that. Of course, there is a requirement for metadata that we have been talking about all along this afternoon. And Ian Fahey is the contact for that. And you can access the information from this link here. And then we are also soliciting for nomination of compound that I talked to you about the metabolite standard synthesis. So anyone who is interested in a particular compound, but it has to be useful to larger community, not just for your lab or just like for 1 or 2 labs. So it should be useful metabolite, but it's not available currently. So you can nominate those compounds. And we have a process for prioritizing for these nominated compounds. I think Susan Sumner is in the audience. She will be able to explain to you if you are interested about how the prioritization is done and how it can be used and how we can you can actually benefit from this resource as such. Of course, I mean, if you contacted this link, Dawn Carter is the contact. She can help you with more information and with the whole process as such. And this is the management structure. This is the workforce behind the screens working and managing the whole program. The leadership flows from NCI and NIDDK collectively. Dr. Philip Smith is actually in the audience. And then we have coordinators from both the institutes, Barbara Spalholz, who could not come here, and Art Castle, actually he's also in the audience. And then we have other, like for various initiatives that I described before, we have a program director for each one of them. And the people that I have put them in the, like a bold, are the program scientists providing scientific input for each one of the RCMRCs that I have described to you before. So, and the rest of the people are providing various levels of advice and input as needed to the whole consortium. So this is the whole gamut of the spectrum, like the the consortium working together and trying to make some progress and hoping that these resources that are put in place will be useful to you, the community itself as such. So I'll stop here and take any questions that you may have.
Got very little time left. I guess 5 o'clock starts. So just, do you want a quick question?
Try to remember where it is now. You put me under pressure. Yeah, you mentioned the bit of text that you quoted when you mentioned about depositing data. You said it was open to anyone that was an NIH grantee or project. Is it open to anyone in the world?
Anyone.
So it's
anyone
anyone
who is interested in the data, provided they can also provide the metadata.
Okay, yeah. So just a quick follow-on question. Would there be any, if someone wanted to submit it to you at the NIH, would it be okay if they also submitted to Metabolites, for example? Is there any, you don't bar people from multiple postings or anything like that?
I don't see any reason why it cannot be posted in multiple places. And actually, there is one activity that is going on with the Metabolites database that they have, that we can have mirror image on both sides, that we can post whatever the data set that they have at DRCC, and the DRCC would be able to host the data. But there are some other considerations for clinical data, because the HIPAA rules are slightly different in the Europe versus in the US. So we are just trying to ease out some of those details. But we really have no restrictions on, the number of places the data can be posted. But this is another one that people should be thinking about. Any other question?
Just the last question, Hunter with that.
Because it's been brought up several times about the exchange, trying to get the exchange between Metabolomics Workbench, Metabolites, and the Australian repositories. Is some of the issue, and sorry, this is going to get a little technical, is it because ISA-tab is RDF-based and not entity attribute value-based to go into a database?
No.
And therefore, the translation from an RDF to an EAV is causing some of the technical difficulties?
There are two different things. The exchange is just very simple metadata exchange. That could be any format. We could do ISA format exchange of metadata, because metadata is easier, but we're not exchanging raw files yet or data files. And the RDF is sort of our part of what Metabolites is doing with ISA team, so that's not part of the exchange yet. But data harmony is sort of a thing that if we think these things in advance, and sometime in future, NIH WordSpread wants to adapt RDF, for example, then actually we have a common basis. You don't have to reinvent the wheel. Okay, is that? We can talk about it later. Anyway,
yeah.
I want to thank speakers for this session, and thank you for all coming. Is there any question, please come and approach us, and we were happy to answer, and thank you for,
Just one thing.
Yes.
Susan, would you like to explain about the prioritization process for the metabolite standards?
If you'd like for me to. Okay, so I'm not sure how I explain about the prioritization, but I'll try. So we would like for people to nominate compounds for synthesis by the Metabolite Standards Core, and you can go to the Metabolomics Workbench site and click on Standards, and that will take you to a page where you can complete your nomination form. When the nomination form is completed, that basically says why you think making the standard is such a good idea, and you can choose to prepare, nominate standards C13 labeled, N15, deuterated, whatever you think is right. You put in all your logic for that. It goes to the executive committee for the NIH Common Fund Metabolomics program. They review the application and then vote on whether or not the compound gets made. And then that really, one of the things that's really important is that the general use of this compound in the metabolomics community, you know, how widely people would want to use it. I think that's a really important part of it. And so, yeah, so that's one of the things that will get it voted in favor if you make that argument really clear, and then it gets distributed. And that's the part that, you know, the amount of the material and how it gets distributed that I think that we're still working out all the details on.
So I've got to finish this, and thank you very much for